Great. Okay, so yeah, my name is Victoria Rachleff. My disclaimer at the beginning, I'm a PhD candidate, nowhere near faculty yet. My advisors are Dirk Keene at the University of Washington and Ed Lean at the Allen Institute for Brain Science. So today I'll be trying to give a pretty high level overview of CAD. It's a NIA U19 funded project. It's a huge project across multiple institutes, the Allen Institute for Brain Science, UW Medicine, Kaiser Permanente, and associated with ACT. The lead PIs are, Ed Lean at the Allen Institute and Dirk Keene at UW. And then just importantly, there's more people than are even pictured here. I'm doing my best to do this justice, but it's a fantastic team. There are a handful of clinicians, of course, neuropathologists, imaging experts, scientists, data analysts, project managers, software engineers. It's huge. And everyone is really fantastic so just wanted to give a shout out to the entire team before diving in. So CAD is a large consortium project, which is really aimed at trying to think about Alzheimer's disease in a kind of a new perspective, less about the proteins and the protein pathology and more about the underlying early cellular and molecular changes. And so, there are some key goals of the project. We want to understand these early cellular and molecular changes. We want, and that's kind of associated with which cells, which specific cell types may or may not be vulnerable early in disease, and understand this in the context of kind of disease heterogeneity and how this changes across multiple brain regions. So, because of the context of this talk, I'm going to try to focus it, on what we do and how we do it, but mainly what we're sharing and how you guys can interact with all of these awesome data sets. So what types of data does CAD generate? There are three kind of major categories, the first of which is quantitative neuropathology. So this happens over at the University of Washington side, and this is tissue processed following rapid autopsy, which is subsequently stained and analyzed. And I should mention all of the tissue used in this project is made possible by brain donation, and it is all post-mortem human brain tissue. So there's quantitative neuropathology on fixed tissue. There's single-cell multiomics on the post-mortem fresh frozen tissue. So that's single-cell ATAC-seq, single-cell RNA-seq, and multiome. And then there's now also a large spatial transcriptomics portion. So just to motivate the project a little bit, probably preaching to the choir in this audience, but as our general population continues to be more aged, Alzheimer's disease and understanding its cause is an increasingly pressing biomedical challenge. There's 50 million people worldwide with dementia, and Alzheimer's disease is the most prevalent form of dementia. In the United States, this comes down to 1 in 9 people over the age of 65. And it's the only leading cause of death that can't be cured, prevented, or slowed. I guess with a little asterisk here, there's some recent advancements that may indicate progress, but there's kind of some caveats to those. And so with the number of people living with Alzheimer's disease expected to double or triple as our population continues to age. So this is a pretty massive, burden, not only the emotional burden on families, but, on the healthcare system as well. So again, this is probably things that this entire audience already knows, so I'll kind of gloss over it. But I mentioned Alzheimer's disease being the most prevalent form of dementia. It presents clinically as changes in thinking and memory, and neuropathologically there are two main hallmarks that people look for, and so that neuropathologists look for. So the first of which is these aggregations of phosphorylated tau in the form of neurofibrillary tangles, and you can see some examples of that here. Probably the easiest to see is this middle image of a mature tangle, where you can see the kind of dark brown staining for one phosphorylated paired helical filament. We actually use AT8, so either of these works, but you can see these massive aggregations of phosphorylated tau within a neuron, and from this kind of characteristic triangle shape, you might guess that this would be a large pyramidal neuron, which is a known cell kind of class that is heavily affected in Alzheimer's disease. And then, there's the very classic and fundamental Alzheimer's protein, amyloid beta, which forms plaques. Without presence of amyloid beta in the brain, there's no neuropathological diagnosis of Alzheimer's disease. So this is essential. However, the relationship between this protein aggregating in the brain and cognitive symptoms doesn't track very well. So this is, increasingly appreciated that this is not the whole story. And then just to make a quick note, dementia is a large umbrella term. Alzheimer's disease is a form of dementia, and there are other types of dementia, vascular, Lewy body, and frontotemporal, with different kind of prevalence in the population. And yeah, just important to note that often these diseases don't happen in isolation. And so there can be kind of, a donor that has multiple proteinopathies. There might be, they have Alzheimer's disease, but you also see Lewy bodies, alpha-synuclein positive Lewy bodies. And so there's just a lot of overlap here. And so it's important to consider that when planning and analyzing these data sets. So kind of the second level of the traditional understanding of Alzheimer's disease is that, yes, we have these two proteins, tau and amyloid, and we know that in most cases, they follow a stereotyped progression throughout the brain. And just very broadly speaking, tau pathology typically begins in limbic structures, it begins subcortically, and ultimately in later stages of the disease spreads out into the neocortex. Whereas amyloid pathology has pretty much the opposite pattern, beginning cortically and moving subcortically in later stages of disease. And, this protein kind of based approach to the disease and understanding the way the proteins move throughout the brain, it's been, the favorite kind of approach to understanding Alzheimer's for about 100 years now, I guess, since the proteins were originally identified. And it's kind of stuck also because there's this pretty clear inverse relationship between cognition and ultimately dementia and increasing pathology in the brain. But, these protein-based approaches have led to limited success, at least in terms of how we are able to treat patients. This is just a recent example. I won't try to pronounce it either, but just to show that there are, some recent treatments that might try to break down these bad protein aggregates in the brain. And, sure, they might be able to, the actual mechanism of action may be working technically, but how much is this really changing the outcome? And of course, just, to mention also, these approaches are not without risk. So that's kind of the asterisk or the caveat of no treatments. There are some, but they leave they leave much to be desired. So this brings us, to this new approach to studying AD that I mentioned in the beginning. And this will get us to kind of the bulk of what we've done in CAD. So it's the idea that, let's think of Alzheimer's disease as a cellular disease. Our brain is made up of individual cells. We now have the ability to really precisely define cell types. So let's try to find which exact cell types are affected most early in disease, and which ones are, and which of those are lost over time. And so this is a slightly, I guess, like in your face image from Google that I pulled, but it links us to our CAD logo here. It's this idea of looking at these individual cells, seeing the affected ones and how that changes across disease progression. So hopefully this paints a memorable picture of our strategy. So I mentioned across key brain regions. I should mention those regions briefly. The kind of first phase of the project, we have based closely off of rock staging. Those are very well studied regions, and we know to expect protein pathology. We're not leading with protein pathology, but we know it's there, and we know that something is happening. And so that's kind of been our guiding direction for the first phase. And so that begins with the entorhinal cortex, hippocampal formation, and then temporal lobe structures. All of the actual data analysis that I'll talk about today and show on our image viewer and our kind of interactive data sets online will be from this middle temporal gyrus. It's a pretty interesting brain region. I mentioned that, tau begins subcortically and moves cortically, and amyloid has basically the exact opposite pattern. The MTG is kind of thought to be this region where the two protein pathologies meet for the first time. And so the idea is that there's probably something interesting happening there. The MTG also marks kind of this cortical transition point between when you kind of have tau plugging along throughout these regions to this kind of explosion of tau throughout the neocortex, and that is associated with pretty severe changes in cognition. So interesting region. It also happens to be studied very in depth at the Allen Institute because it also is the region that is often removed in temporal lobe epilepsy surgeries. And so neurotypical tissue from the human brain has been really well characterized. So there's just a lot of reasons that this was the starting point. This is a huge project with ultimately is going to profile, I believe, 30 brain regions. But that's just our motivation for why this was the first. And then, you know, as I mentioned in the beginning, we're going to then describe our quantitative neuropathology, our single cell multiomics, and spatial transcriptomics on the MTG. So just to kind of give a little more background on this, because if I jumped us straight to the image viewer, it might not, you know, unless you already had a really specific idea and question in mind, it might not make sense. So let's walk through this workflow a little bit. These are tissues from donors that have, you know, postmortem donors that have undergone rapid autopsy. They are sectioned at 5 microns and stained for some key markers to identify major cell types, neurons, microglia, and astrocytes, as well as key pathologies. So phosphorylated tau, amyloid beta plaques, TDP43, and alpha-synuclein. They then undergo whole slide imaging, and then a team in Dirk Keen's lab goes through and does just super detailed, really precise kind of image processing workflow where they're able to identify the cortical layers, layers 1 through 6, and within those cortical layers, they're then able to apply masks to quantify any number of a pretty impressive number, I should say, of relevant features related to these individual stains. So you can, you know, it's important to look at maybe this, in a lot of ways, the disease in a more refined way. And so one of those ways is looking at things in a layer specific manner. And then within layers, you can start to ask really specific questions about microglia branch length or what have you. So yes, this is the workflow, you know, just for this first region. It's huge, you know, huge effort. There's been 720 slides. There's, you know, 7 stains. Some of these are duplex, as they're mentioned here. There's the cortical layer specification and the masking. So it's a huge effort. And there's a super cool, what we kind of are now calling like a virtual microscope online that allows you to look at these whole slide images, zoom in on them, and see the annotations as well as the masking, which I'll show in a second. So this is perhaps going into a tiny bit too much detail, but yeah, I'll go through it relatively quickly. So first stain, as an example, is AT8 phosphorylated tau. You would see just the typical, you know, stain here. And then where you see overlays in red and green are the masking for showing what the computer is essentially picking up as positive regions. So for each stain, there's a core set of features that are extracted. So there's the percent area. So there's the percent of the tissue that is stained for that marker. There's the number of cells in a given region. So a number of cells normalized per area. There's the average size of a cell that contains this pathology, as well as in a duplex stain, the percent of area that is co-expressing two markers. So you can kind of expand this to the entire set, as I mentioned, with the addition in blue here of stain-specific kind of special categories. So, you know, with plaques, might be really interesting and important to, at a highly quantitative kind of level, look at dense versus fibrillar versus diffuse plaques. To my knowledge, like a survey of a large number of donors with highly quantitative methods like we use of these kind of differences has, you know, it's not super common. And so this is a really cool data set within and of itself. Then, you know, there's hematoxylin, which is, you know, sure, not the most exciting, but there's some features, you know, related to the nucleus. And then, you know, yes, so astrocytes, we might be interested in branch area and length, microglia processes. I forgot to mention here, but there's also an aspect of this that can kind of look at the roundness and identify whether they're activated or inactivated microglia. And then there's an interesting finding as well related to neurons stated by NUN that at least with this specific neuronal marker, NUN, We see an interesting decrease in NUN expression with increasing pathology. And it's a really strong relationship. And it's not that the cells are being lost, it's that they're specifically not expressing this marker as well. And so a lot to be unpacked there, but kind of an interesting, unexpected finding. So now that we understand all the features, which are summarized here, we can get into a little bit of the basics of what we found with them. So this is just looking at, again, this is all middle temporal gyrus. This is split into the 6 cortical layers for analysis purposes. Layers 5 and 6 are combined and stacked or split by Brock stage. So, you know, as a start, this was like one of the first plots we made, right? Just as a sanity check of, you know, across the layers, we would expect that as you increase Brock stage, you're increasing the amount of phosphorylated tau positive cells. And we do. The kind of more interesting thing is that within each of these stages, especially the stages, you know, Brock stage 4 and up are where you really expect to see tau pathology in the middle temporal lobe. And so you start to see how much variation there is within these discrete stages. And the way that neuropathologists do this right now, it's a Brock stage 4 is a Brock stage 4. There's no note saying it's a Brock stage 4 high, medium, or low, let alone a Brock stage 4 numerical value. And so this just shows that, you know, there's a whole lot of variation within each individual clinical stage or neuropathological stage that can be unpacked and studied and is probably relevant and interesting to understanding the disease. So that's kind of the takeaway from this plot here. And then, you know, there's a ton of features, right? So probably looking at them one by one in our box plots are not, you're not going to find everything. And so we started with some like semi-simple approaches like doing PCA, and on this axis here, you can see ordered list of features from PC1, and don't expect you to read them, which is why I made the boxes in the notes, but you can essentially see that there are some key changes that are immediately kind of popping out at us. Like I mentioned, this decrease in neurons, and then, you know, actually my computer's blocking it, but I should have it memorized by now, increased in amyloid, microglia activation, increase in tau. And so, you know, these were kind of the sanity check level things. And then what some biostatisticians in the group did to take this a whole nother level, and I've attempted to describe this in multiple talks now. I have a detailed description and a link to our manuscript. If you actually want to know what goes into this, I will send you that, and I won't attempt to describe it, especially on a recorded talk. But the idea, very, very simply put, is you feed in most of these features, and basically they've developed a continuous model, and that will kind of rank all of our donors across kind of a numerical scale. And so this would be like, you know, donor 1, each bar, donor 2, 3, 4, all through 84. And you can see red is higher, blue is lower. And so you can basically see as you get more affected on this pseudo progression score, you have more amyloid, you have more tau, you have fewer neurons, or at least fewer NUN labeled neurons, and lower cognition. So this is a really neat thing because, as I mentioned here, there's a lot, again, left to be desired with this kind of categorical Brock staging. We see the variation within each stage, and this allows us to go from a categorical approach not only to like a semi-quantitative, but to a fully quantitative, like continuous approach to understanding donors' kind of full neuropathological kind of stamp, if so to say. So that I'll leave it for there with the neuropathology and how it leads to the pseudoprogression score. I'll jump to the Omics, and then I'll show the data and how to navigate our website. I have a few, I'm okay-ish on time. So most people are, you know, familiar with this process now of a general single nucleus omics workflow. This is the MTG shown here. It will be dissected out, and then they will isolate nuclei. Then they use 10x. So this would go through like a droplet-based single nucleus RNA sequencing approach where every single mRNA transcript within it is, you know, barcoded within a given cell and then sequenced. And so this allows you to go from kind of this broad approach of like, oh yeah, there's a triangular looking pyramidal neuron labeled with tau, to all of a sudden there's, you know, this extremely rich classification of really refined cell types. And so this is just an example of kind of a cell type wheel of what level of resolution doing single nucleus omics on brain tissue gives you. I mentioned because there is also the ability to study the same brain region in surgically resected tissue. There is really fantastic patch-seq data that allows us to also look at these cells' morphoelectric features and kind of, look at their morphologies here. And so we can see when we have, our disease data, which cell types they are, what they look like, which cortical layer, we think they would belong in. And then of course we can ultimately check that against spatial transcriptomics. And so this is a pretty powerful tool, and that's kind of what we're hoping kind of the cellular approach to studying AD and taking advantage of the Allen Institute's strengths will really give to this data set. So what the kind of analysis team, Kyle Travaglini at the Allen Institute has done is map the data, the single nucleus data from these disease donors onto the really well characterized Allen reference data, which is also from the MTG. So you're able to do this. And this is just showing the kind of broad categories of glia, excitatory and inhibitory neurons. And then this is just showing if you kind of overlay Alzheimer's disease neuropathologic change, which captures Brock-Fell and CIRAD kind of all into one, just to give a snapshot of general disease stage. You can kind of already see some interesting points within this data set. So these are our results, and then I'll switch to the website and data tools. So what we were really interested in knowing, I mentioned in the beginning, is which cell types are affected, which specific cell types are affected in disease, and in what way. Are they increasing or decreasing in the brain? And this is basically looking at change across disease progression. These are kind of the effect size. And basically you can just say, like, if the bar is going down, there's a decreased relative abundance of this specific cell type. And if the bar is going up, there's an increased relative abundance. And so what pops out, at least to me, are these green ones here. These are layer 2-3 intratelencephalic projecting neurons. Just to link this back to the kind of classical Alzheimer's disease literature, this is where those large triangular pyramidal cells that accumulate high levels of phosphorylated tau are located. And so this is, really interesting, but not totally unexpected finding. And then you see some pretty obvious increases in microglia, one particular population of microglia and astrocytes, coming to the site. So interesting and not totally unexpected findings. What is a little more unexpected is that there's these somatostatin positive interneurons that are showing a pretty kind of slightly more broad and pronounced decrease in relative abundance. And so we wanted to understand this a little bit more and how these two changes might be connected. With cortical excitatory neurons, we know enough from the single nucleus RNA sequencing data already to be able to say they're layer 2-3 IT and define them that way based on just the RNA profile. Whereas that's not quite the case with somatostatin positive interneurons. They're more migratory, right? So what we were able to then do with spatial transcriptomics, oops, didn't mean to jump ahead there. What we can then do with spatial transcriptomics is say, okay, well, we know that these are the types that are the most affected. Let's look at them in situ and see which cortical layer they reside in. So the darker yellow, I guess that color is, are the ones that are the affected cell types. And those preferentially are located in layer 2-3, which, as I mentioned, is where our affected excitatory neurons are. So this is a really, really interesting connection, because then it at least gets us, our two main findings are localized in the same layer. It's a layer that we also know has pretty high amounts of protein pathology. So we're not ignoring the protein. We're just taking this approach and then considering, that in light of the protein pathology. And so the final step here, the coolest part, and I guess the smallest plot, apologies, is to understand, okay, we now know what is lost, but when. And that's where the pseudoprogression score comes in. And basically just, this is like the zero, the lowest pseudoprogression. So this would be like a brain without any pathology or disease, and this would be a brain with the highest pathology and disease. And so if you look across kind of that low to high spectrum, and you look, let's see, at this green bar, you can see that it's pretty consistent, and then there's this drop off, let's say like late in pseudoprogression. Those are our layer 2-3 IT types. So basically you at this kind of, let's say 0.7 or 0.8 stage, there's a steep drop off, which we also know is when the level of pathologic proteins really ramp up. So that's interesting, and again, not unexpected. And then you see you see pretty consistent increases in these non-neuronal cells starting earlier in this disease pseudoprogression. This orange-ish bar here marks the somatostatin positive interneurons. And what I think is the most interesting here, and has not really been shown otherwise, is that the changes in this cell type start early in disease, before protein pathology has really become prominent. And so that's an early affected cell type with early molecular changes that we would be very interested in. So this kind of summarizes how we get the data, what the data looks like, and what we internally have been able to extract from it as it relates to understanding Alzheimer's disease. So, there's been already pretty good uptake of this data set once it was released. There was an interesting NPR piece, and it's already been downloaded by 100,000 of our data files have already been downloaded. So I don't know if that necessarily means 100,000 users, but a lot of our files have been downloaded. So that is great. So at this point in time, 118, I will jump us then to the interactive part. So hopefully everyone can still see my screen. This should be the second link that I sent over. This is our homepage for CAD. There's a blurb on what we're doing, which you all know now in depth. And there's a few ways to explore the different data modalities. So there's looking at the cell type data from the single nucleus Omics. There's the neuropathology and donor index. There's epigenetic data to look at the kind of the peaks. And then there is, if you want to have more of a hands-on, if you don't want to download the raw data, but you want more of a hands-on interaction with the single nucleus Omics data, then we offer this cell by gene tool, which is, for those who have used it, it's super, super cool and really powerful. So anyways, that's the overview. If anyone has any questions so far, when I started presenting my screen, the Zoom window split into 5 things. So I can try to pull back the chat, but otherwise I'll go into just a slightly more detail for the different categories.
There are no questions yet in the chat.
Okay.
So why don't you go ahead with this, and we can do questions at the end.
Okay, perfect. Okay, so just to show this comparative viewer. So this is the reference taxonomy for the MTG. So this is what is in essentially healthy tissue. Then this is our Alzheimer's disease taxonomy. So if you're kind of used to looking at these UMaps, you can pick out your islands with your cell types of interest. You can see these are more dense, and that's because we have already sampled more individual nuclei than the reference itself. I showed a screenshot of something like this, and so this is kind of that same UMap, and you can pseudo color it by not any number, by these pre-selected features of interest. So, it's good. I forgot to reload this before the talk, so maybe I should have done that, the whole page. But anyways, you should be able to kind of overlay any of that. Oh my goodness. Metadata. Wow, okay. Well, maybe we will jump to a different viewer, given that this one is not cooperating at the moment. Well, now we can see the one thing I asked to see, we can see really well. Everything else has disappeared. There's another kind of interesting thing you can do, which is kind of looking at these dot plots for each of the cell types. And wow, that's super interesting that it works this way. I haven't actually used this part of the tool very much, so apologies. Yeah, but basically you can look at some kind of simple high-level features. You can also click this button at the top that allows you to view disease progression, which is our pseudo progression. And so whatever gene or cell type you had selected up front, it will show you where that falls along our pseudo progression score. So that is a new feature based on some interest. So that is a cool button to click. I will then jump to our donor metadata. So this is probably, if you're interested in like looking at this data set and you want to understand the first step, which is like, what are the characteristics of these donors? You'd want to start here. The filtering tools are pretty nice, actually. So you, these are kind of like bar plots within overlaid. So it shows you kind of what the distributions are like without having to really do anything. You can select any of these features to subset the list, and that will immediately show up down here. And then took me a second to realize this. If you have a feature that you're like, why didn't they show late stage? All you have to do is click on the settings and add that, and it will appear on your as a new column. So yeah, you only want ApoE4 people with dementia, high ADNC. So, this type of thing. Okay, now we're down to 14 out of the 84. So I use this tool actually all the time. For my thesis work, we were only able to do it on a subset of the cohort. And so I wanted to be very careful about the donors I selected. And so this was a really nice tool to use for that. And then finally, there's this kind of virtual microscope that I mentioned, and I'll only spend, 1 minute, I'll be done. And so you can select any donor that you want to look at. This happens to be a 90-plus-year-old female with ApoE4, pretty high pathology. And so I guess I'm using my mouse, but you can also use the buttons. You can zoom in, you can see the cortical layers, you can see what the tissue itself looks like, and then you can see what the masking picks up. So this is actually also really useful if you're going into to even use the omics data and you want to understand what your donors look like from a neuropathological perspective. This is it's really cool. And you importantly can go through and select where now that was like, I guess that was 60, 10 in ABL1, makes sense. You can select any of the stains and do the exact same thing. And just to end here, pretty much everything can be downloaded directly at, and even stuff I didn't mention, like MRI volumetrics, Luminex data, there's more, can pretty much all be downloaded if you want to work with kind of the slightly more raw forms of the data. So with that, I will find my mouse on one of my screens and stop sharing. And I can take any questions as well.
Great. Thanks, Victoria. If folks have questions, go ahead and ask them. There was one already. Let's see. Eric, do you want to just ask it out loud?
Sure. Thank you for the great talk. I was just really curious if you have a way of looking at people with this sort of middle diagnosis of mild cognitive impairment.
Unmuted. There we go. A way, sort of. So only in the sense that we have, I believe off the top of my head, 2 donors where next to their dementia note, it says /MCI. So not nearly powered to do anything. And I don't believe those particular donors stood out in any interesting way. I guess this could be a plug, though, that the next phase of CAD is really interested in capturing more of the early stage donors, as well as different, capturing more polypathologies and all of those other things. So at this point in time, not really. Technically, 2 donors meet that criteria, and we don't know they haven't stood out or caused any issues in our analysis yet, but that's about it.
Other questions? Generally, Victoria, I'll toss one out. So you said 100 or 100,000 downloads of the data. What do people use that data for?
I hope and imagine, and maybe this is like a very selfish perspective. I'm imagining graduate students in a lab that perhaps can't generate this large of a data set, and they have a specific question they want to ask, and we're covering enough brain regions that hopefully if they're in a cerebellum lab or they're in different groups, they can say, oh, that's my brain region. They can go to the data set, download it, and check either their results to the data, or perhaps even just start something from scratch. Also, people, that are doing things more specific, like they're really interested in a specific gene, they can cross-reference that pretty easily with our data set. So I could see people coming in to just like do those types of like gene checks, I could see people coming in to use it to compare it to their single nucleus omics data, or hopefully graduate students like myself using it to generate hypotheses. But I don't know how we get that type of feedback, but I would imagine the Allen Institute does. But that would be interesting.
Yeah, it's worth noting with prior Allen data sets, it is common for investigators and trainees to use those data and include them in it either as an adjunct to existing studies. So, for example, if you're doing a study in a rodent model, evaluating a particular gene product and its role in AD-related pathology, it's pretty straightforward to download data from a data set like this, to analyze it just like you would any data and to use it in your publication. That's definitely permissible. Our group used some earlier Allen data, and we had a sort of a hackathon based out of the old dementia, aging, and TBI study that they published a bunch of years back. And three of our students published 3 different papers out of that just data set that was essentially only that data. So this is something that you can incorporate into your ongoing studies, not just for hypothesis generation, but actually for something you could report.
And I guess I'll just add, that's a great point. And to add one thing to that, as the data sets get bigger and the analysis gets much more complex, it makes it much less accessible for someone without computer science background to do these things. Like this data set, I would say almost certainly falls in that category. So we've kind of tried to make both the visualization tools and the download options for basically like a non-coding user, a basic user, and like a super user. And so, there should be a way to like hopefully get all the information you want without typing a single line of code. And then, for the very few that are going to like properly merge this with their other large data set, they can get the raw files. So there should be every stage for every kind of level of expertise.
So there's a question in the chat says, can you talk about the representativeness of the CAD data? For example, are samples representative of the general population or high-risk ADRD? How are donors recruited?
So it's a Seattle-based cohort. It was done like and there's probably a proper word for this in the field, but basically like on a start date, it was all subsequent donors that came through rapid autopsy were included. So with the exception of excluding genetic, like if the person had Down syndrome or some things that we knew would not make this a kind of a simple sporadic Alzheimer's disease data set, those genetic conditions were excluded. But it is representative of the Seattle population. It is skewed towards higher pathology, certainly. We have both ACT. So I don't know if I can get too much into the like, I don't know if I know enough to say too much of the specifics beyond that of like the breakdown of who is in ACT versus other studies.
Yeah, my understanding is this is recruited entirely out of the ACT cohort. Is that right? I
thought that there were additional, but given that is the way we also put the logos on the front, it could be that this is exclusively ACT. That's very.
So for those that are new, probably another AD at UW will talk about the ACT cohort, which is if you're in this environment is a really important piece of this environment. So that's a Seattle-based longitudinal cohort study that is community-based, which is valuable, but it is community-based out of the formerly the group health, but now the Kaiser HMO, right? So that is not representative in the sense that it is restricted to people who are within that healthcare system, which are people with healthcare insurance, right? And so that does include a selection element to it.
And I will just add one note on the cohort itself too, that I mentioned the Seattle-based cohort. We have not a lot of representation of like non-white individuals. And so a huge thing with the renewal, which will hopefully we're in that kind of renewal phase right now, will be expanding to not only be processing donors from Seattle, but other states and locations, other neuropathology cores, to hopefully really change that picture and be more representative of the larger population.
Don, you turned on your camera.
Yeah. Super interesting. And it seems like that if these subjects are from ACT, that they would also be in the ADRC database. So you would have cognitive scores, across time as well. Is there some limit to sort of using both databases for people who are in the ADRC at UW? Like, seems like if you were to link the two, you would tend to identify them, but I guess
so I'm trying, like, I know we have, for example, CASI scores, and I don't know which that is associated primarily with.
Yeah. So that would, so I think that would be something to reach out to Dirk.
Yes, certainly.
And
Yeah.
Paul Crane and Ed Lean, the three of them together. So because these are ACT participants, yes, there is longitudinal cognitive data. They aren't formally a part of the ADRC. ACT is not synonymous with the ADRC, but there's a close alignment. But ACT does it. It's a cognitive aging study, right? And so there probably is those data. It is. I don't think it is linked through CAD, but in principle, there is probably an opportunity through collaboration to do that.